All right, now we're going to talk about some of the deductions that Mendel was able to make as a result of his very structured experimental process and how he counted all of the offspring of all of his different plant crosses and really documented all of his data very, very well. And so he was able to come to some conclusions. Excuse me. On page 221 of your textbook is a section titled Mendel's Model. And it goes through 4 deductions that Mendel was able to come up with. Essentially, the idea that every trait had 2 what he called factors, we now call them alleles, and that not only were there 2 alleles per trait, but that there could be different versions of those factors. And again, we call those alleles. He also deduced that each individual inherited 1 factor per parent, and that when an individual mated and had offspring, that the parents' 2 factors or alleles per trait segregated independently into the cells that were responsible for creating offspring. So let me kind of dissect that for you just a little bit. So when it comes to the law of segregation, what Mendel was deducing is what we talked about last chapter with meiosis, okay? Remember when we're talking about meiosis, we're talking about a diploid germline cell that undergoes 2 rounds of cell division to produce 4 haploid daughter cells. And in organisms that reproduce sexually, meiosis is producing gametes. So each of these gametes is haploid. Well, the original parent cell started off as diploid, and so the 2 different chromosomes, the homologous chromosomes, they segregated or sorted independently into the daughter cells. Okay, so Mendel's law of segregation is the same thing as independent assortment in meiosis. All right, so how can we use Punnett squares to predict the offspring of particular genetic crosses? All right, so when we're talking about offspring, we are talking about an egg cell, which is haploid, crossing with a sperm cell, which is also haploid, to produce a diploid zygote or fertilized egg cell. Okay, so when we set up Punnett squares, if we're looking at 1 trait at a time, these are called monohybrid crosses. So if we look at this Punnett square in this figure, this is a cross of 2 heterozygotes, okay? Mom and dad. Well, when mom and dad undergo meiosis, remember, these 2 alleles are on the homologous pair of chromosomes, okay? So when mom and dad undergo meiosis, each of these alleles is going to sort independently into gametes. Okay, so mom's egg cells will either have a dominant P allele or a recessive P allele. Dad's sperm cells will either have a dominant P allele or a recessive P allele. Those are the only possibilities with respect to the P gene, the P color gene, for these two parents. And so when we set up our Punnett square, we have to put the gamete possibilities for one parent down the side and the other parent across the top. Okay? Well, when fertilization happens, an egg and a sperm have to come together. So we could get an offspring from that egg and that sperm. The offspring would be homozygous dominant. Or we could get an offspring from this combination. That offspring would be heterozygous. Okay, so for the cross of two heterozygotes, we would expect in our offspring population one homozygous dominant, one homozygous recessive, and two heterozygotes out of every 4 offspring. Okay, so let's do another example. Let's just assume that we're talking about human beings and whether or not you can roll your tongue. Okay, so let's say that dad is heterozygous for tongue rolling and mom is homozygous recessive. She cannot roll her tongue. Okay, well, the gametes from mom and dad can only have one R per sex cell, right? Because the gametes are haploid. So if we do the Punnett square for this cross, let's put dad's gamete possibilities on the top. Dad can donate big R or dad can donate little r. Mom can only donate little r because she's homozygous recessive. So for the offspring, we could expect to have this offspring to be heterozygous. So dad's big R comes down, mom's little r comes over. Okay? For our top right box, dad's little r comes down, mom's little r comes over. For the bottom left, dad's big R comes down, mom's little r comes over. For the bottom right, mom's little r comes down, dad's little r comes down, mom's little r comes over. Okay, so this couple would expect that 50% of their offspring would be able to roll their tongue. Okay, our phenotype ratio would be 2 dominant phenotypes to 2 recessive phenotypes, but ratios are kind of like fractions. You always express them in lowest terms. So more correctly, we would say there's a 1-to-1 ratio for dominant phenotypes to recessive phenotypes. So this couple would have a 50-50 chance as to whether or not their child could roll the tongue. Okay, well, let me just clear up one potentially common misconception. Let's assume that mom and dad's first child can roll their tongue. Does that mean that if they have another child, They have a 1-to-2 ratio. In other words, they're twice as likely for the next child to have a child that cannot roll their tongue. The answer is no. Okay? Each fertilization event is independent. Data in independent. Okay, so just because baby number 1 could roll its tongue, when it's time for baby number 2 to come along, they're going to have all of these same possibilities for baby number 2. Okay, has nothing to do. Baby number 1's genotype is going to have nothing to do with baby number 2's genotype because every fertilization event is independent. Okay, let's do another one. Let's do, for monohybrid crosses, it gets pretty redundant pretty quickly. Let's go back to our peas and let's talk about yellow peas, which were dominant. Yellow is dominant, crossed with green peas, which was recessive. Okay, so let's have, actually, I'm going to change that because that's just not very interesting. Let's make it different than what we did with the tongue rolling. So let's have a true breeding yellow. Oops. Crossed with a heterozygous yellow. Okay, so we put 1 gamete possibilities across the top and 1 parent's gamete possibilities down the side. Now, Y's are some of those letters that the big Y's and the little Y's are going to look alike unless you're really careful. Okay, so if we cross a true breeding yellow plant with a heterozygous yellow plant, then all of our offspring are going to be yellow because the parent that is homozygous dominant for yellow, the only allele that that parent can donate to the offspring is the dominant allele. So every child is, or every offspring is going to get at least 1 big Y. Well, if every offspring has at least 1 big Y, then the dominant phenotype is going to be expressed. Okay, so we're going to have 100% of these offspring be yellow. Okay, well, Mendel next wanted to look at pairs of traits in combination with each other. So in other words, the true breeding strains of pea plants that had yellow flowers, yellow peas, typically had peas that had a round texture. And the recessive green peas were usually wrinkled. So he wanted to know, did round and yellow always have to show up together, and did green and wrinkled always have to show up together, or could there be yellow wrinkled and green round peas? Okay, so again, starting with true breeding varieties of each parent, okay, yellow round times green wrinkled. Now remember, you use the same letter for each trait. So because yellow is dominant, we're using big Y for yellow and little y for green. We don't change the little y to a G. That would be too confusing. Okay. All right, so the F1 generation, you can already surmise, is going to be 100% yellow round peas because all of the offspring are going to be heterozygous. But what about the F2 generation? What when we cross those two heterozygotes together? Well, this is a dihybrid cross because we are interested in two traits at a time. So the first thing we've got to do is we've got to figure out, well, what are the gamete possibilities? When the homologous pairs of chromosomes sort into the gametes, how are they going to sort? Okay, well, I want to draw it like this for just a minute. Okay, our Ys are on a homologous pair of chromosomes. Our Rs are on a homologous pair of chromosomes. So, if during metaphase 1 our homologous chromosomes lined up like this, then we would get gametes that have the big Y and big R, and gametes that have the little y and the little r. But if the homologous chromosomes lined up like this, we would have gametes that have big Y, little r, little y, big R. Okay, so for dihybrid crosses, we're going to have 4 possibilities for how the alleles can sort into the gametes. So when we set up our Punnett square, we're going to have a bigger Punnett square. It's going to have to be a 4 by 4 square. Okay, I don't want you to worry about this. This is just a hypothetical example of if there was an independent assortment. I just want you to look at how the Punnett square is set up. Okay, so for each parent, we could have big Y, big R, big Y, little r, little y, big R, little y, little r. Okay, now I want you to pay attention to how the genotypes are written for each potential offspring. Okay, so this top left offspring is going to get big Y from dad and big Y from mom. Big R from dad and big R from mom. Notice when you write the genotypes, the 2 Y's are always written together side by side and the 2 R's are always written together. If there is a dominant allele in the genotype, it is always written first. So for this top left offspring, you would not write the genotype big Y, big R, big Y, big R, because YR is not a genotype for a certain trait. Okay, you would have to put big Y together. That's going to tell you the pea plant or the pea is yellow. The big R is going to tell you the pea is round. Okay, all right. So when you do these dihybrid crosses of the F1 generation, then what you see is that you're going to have most of your offspring with the dominant phenotype for both traits. Most of them, more than half of them, are going to be round and yellow. But you will get some of them that are yellow and wrinkled. Well, what do yellow wrinkled peas, what do their genotypes have to be? They have to have at least one big Y in order to be yellow, but they have to have 2 little r's in order to be wrinkled. Okay, so here are those 3 possible genotypes. Okay, we're also going to have some green round peas. If they're going to be green, they're going to have 2 little y's. If they're going to be round, they have to have at least 1 dominant R. Okay, so here we've got those 3. And then we do have some that are, or 1 out of 16, that are green and wrinkled. In order to be green and wrinkled, homozygous recessive for both traits, they've got to have 2 recessive alleles for each trait. Okay? All right, so let's practice, technical difficulties. let's practice a dihybrid cross. Uh-oh. Okay, well, I'm not sure why it won't let me erase right now. Let's practice a dihybrid cross with purple flowers, flower color, and pea color. Okay, so let's say we've got heterozygous purple flowers with green peas crossed with white flowers and yellow peas. Okay, what is that cross going to look like? Okay. So for our purple flowered plant with green peas, big P can sort with that little y, or big P can sort with that little y. Little p can sort with that little y, or little p can sort with that little y. Okay, for parent number 2, little p can sort with big Y. Little p can sort with little y. Little p can sort with big Y. Or little p can sort with little y. So these are the gamete possibilities. Now I want you to pause the video, try to fill out the Punnett square, then come back and check your work. Okay? So I'm going to go ahead Okay, so here's the completed Punnett square. So now, how many of our plants are going to have purple flowers and yellow peas? Purple and yellow. Okay, purple and yellow, purple and yellow, purple and yellow, purple and yellow. Okay, so we're going to have 4 plants that are purple and yellow. How many are going to be purple and green? Purple and green, purple and green, purple and green, purple and green. All right. How many are going to be white and yellow? White and yellow, white and yellow, white and yellow, white and yellow. 4. How many are going to be white and green? White and green, white and green, white and green, white and green. 4. Now, you can check yourself. Do your counts 4 plus 4 plus 4 plus 4, do they add up to 16? Yes. So we did this Punnett square correctly. So the point of all of this is that when we've got dihybrid crosses, as long as the alleles for the two different traits are on two different chromosomes, then they are going to sort independently